Today we're going to talk about drug receptors and their importance. In this presentation, we intend to cover what are receptors. We're going to look at the definition. Why are they important? How are they discovered? So that is our learning objectives. We're going to look at what are receptors, why are they important, and how are they discovered. So let's consider a few drugs. Say captopril, atenolol, diltiazem, omeprazole, amiodarone, cyclophosphamide. Let's write their mechanism of action. So captopril, we all know it's an ACE inhibitor. Okay, what is ACE? It is angiotensin converting enzyme. What is atenolol? It is a beta blocker. Specifically, a beta 1 antagonist. What is diltiazem? It's a CCB. What is a CCB? It's a calcium channel blocker. It is omeprazole. It's a proton pump inhibitor. Proton pump inhibitor. It is amiodarone. It is an anti-arrhythmic drug. But here we are looking at its role as a PGP inhibitor. What is PGP? P glycoprotein. What is cyclophosphamide? It is an cancel drug. It acts on the DNA. This mechanism of action of these 6 drugs lead us to a generalization. So we can say drugs, they act on something and that interaction results in a response, and that response translates into a therapeutic effect. So this something on which the drug acts, we call it a receptor. Okay, there's something on which the drug acts, we call it a receptor. Okay. So let's ask ourselves the question, what is a receptor chemically? This is a point that we are going to consider. What is a receptor chemically? Let us look at our discussion which we had previously. Let's consider the mechanism of action of these drugs. So captopril is an ACE inhibitor. We said ACE refers to angiotensin converting enzyme. Now all enzymes are proteins. So captopril acts on a protein. Atenolol acts on a beta blocker. So it's a beta blocker. It acts on the beta receptor. So what is this beta receptor? The beta 1 receptor. It is again a protein. Diltiazem acts on the calcium channel blocker. What is this calcium channel blocker? It is again a protein. Omeprazole acts on the proton pump. What is this proton pump again? It's a protein. Amiodarone acts as a P-glycoprotein inhibitor. So here again it is a protein. Cyclophosphamide acts on the DNA. Okay, so we say from this small discussion that we have drugs acting on proteins and we have drugs acting on DNA. Okay, so from this, let's ask ourselves the question, what are proteins and DNA? So they are basically macromolecules. So what is a receptor chemically? We have said that the receptor is basically macromolecules. In the examples we cited, we found the macromolecules to be proteins or the nucleic acid. So then, with this information, we can try to define a receptor. So we say receptors are macromolecules What macromolecules? They may be proteins, they may be nucleic acids, they may be carbohydrates, or they may be lipids. Okay. That are present on or within the cell. Why this cell? Because cell is the structural and functional unit of the body. Okay, which means if it is the functional unit of the body, if you can interfere with some function of the cell, then ultimately you interfere with the function of the body. That's the idea. Within the cell, that brings about a response which translates to a therapeutic okay. So with this we have defined what a receptor is. Receptors are macromolecules present on. When we say on, we mean they may be extracellular, within, they may be intracellular. Okay, so which they may be extracellular or intracellular. Upon binding a drug, they produce a response which translates to a therapeutic effect. So that is the definition of a receptor. Now, receptors are drug targets. Okay, these receptors are drug targets. Okay. Right, so we need to refine our understanding a little bit more. Now we said that they are present on or within the cell. The cell needn't be within our body. It can also be present on pathogens. These pathogens may be microorganisms. Organisms like bacteria, fungi, viruses. So we have antibacterial drugs. We have antifungal drugs. We have antiviral drugs. They can also be parasites, like worms. So we have anti-parasitic drugs. Okay. Or they can even be present on insects. which case we have insecticides. Okay. So the receptor needn't be present on the body, they can also be present on. So receptors can be present on pathogens. Okay. So now we have defined receptors as macromolecules which are present on or within the cell, and the cell may be a human cell or it may be a pathogenic cell. But when the drug acts on these receptors, they produce a response that translates to a beneficial effect, which we call the therapeutic effect. Okay. So now we're going to say the next point that we're going to take up is what is the importance of drug targets. Okay, so in our previous discussion, we said that receptors are drug targets. Now we are going to ask the question, what is the importance of drug targets? Now, there have been two main approaches to discover novel, two main approaches to discover or invent novel drug molecules. And what are these two major approaches? Two major approaches have been the traditional, it's also called classical, it's also called irrational drug discovery forward. So they have been referred by different names, but they have been called traditional or classical approaches, irrational drug discovery, forward pharmacological approach, etc. And the second one is called rational drug discovery or reverse pharmacological approach or modern drug discovery. Okay. In the traditional or classical approach, we have a drug and we try to find its target. Why do we need to find the target? Because we can understand the mechanism of action and then we can modify the drug to get a better drug. In rational drug discovery, or this is also called target-based drug discovery, here the target is required to invent a new drug. Okay. So in the first case, we have the drug itself. So this is also called ligand-based drug discovery. Okay. But what is common to both this is that the target is required. Okay. So this is the importance of drug target. Okay. So this is the importance of drug target. Okay. Now we ask ourselves the question, how do we discover a drug target? Okay. Here we are going to discuss the modern approach. By modern approach, we mean the post-genomic approach. By post-genomic approach, we mean after the publication of the human genome. Sequence. Okay. So there are many modern approaches, but we will try to explore it with the help of an example. Let us consider a case of maybe cancer. Okay. So imagine there are two friends. Okay. So we have friend 1 and friend 2. Friend 1 has got cancer, and friend 2 doesn't have cancer. Okay. Let's say the nature of cancer is that it is a solid tumor. Okay. Meaning biopsies can be done. Biopsies are feasible. Okay, so the cancer has advanced enough for us to do biopsy. Okay, so this person obviously doesn't have a tumor. Okay, so let us say this tumor is in a muscle. Okay, that is easily accessible. Okay, for the sake of our discussion, let's say it's in a muscle. Okay, so we have got these two friends. One has got cancer, one doesn't have cancer. Okay. So, first thing what we do is we take biopsies of both. Okay. So we take a biopsy from friend 1 and we take a biopsy from friend 2. So from friend 1 we do a biopsy. In friend 2 we do biopsy. So what is biopsy? Biopsy basically is a removal of a small sample. so biopsy is done. So let us draw a tube. Okay, this is F1's tube, and this is a biopsy sample. Okay, so this gentleman, let's say his sample is this. This is F2. So biopsy samples are taken. So these biopsy samples, from both messenger RNA is extracted. Okay. Messenger RNA is extracted. So there are different protocols how to extract messenger RNA. We won't go into the experimental protocol, but there is a method by which we can extract messenger RNA from tissue samples. So messenger RNA is extracted. And this extracted messenger RNA is subjected to an enzyme called reverse transcriptase. Now, reverse transcriptase converts messenger RNA into complementary DNA. Okay, so now we have got two complementary DNAs for F1, and we have got a complementary DNA for F2. Okay, so we have complementary DNA. Then we take DNA chip. Okay, DNA chip, which is also called a microarray. Okay, so what is a microarray? So let us discuss what is a microarray. So to make a microarray, we take genomic DNA, okay. And then we get primers for each gene. Okay, there are about 23,000 genes. Okay, so for each gene we have primers, and individually we do PCR for each of the 23,000 genes. Okay, so we do 23,000 PCRs. From each PCR we get one gene. Okay, meaning we get a double-stranded DNA. Double-stranded DNA for each gene. Now, if we denature this double-stranded DNA, we get a single-stranded DNA. So, in this DNA chip, which is basically a glass slide with a small square, which is having different locations. Now, at each location, the single-stranded DNA from one gene is put there. The next spot will be occupied by the single-stranded DNA from the second gene, and so on. Okay, so when we spot 23,000 genes, then that becomes a DNA microarray. Okay, so this is a microarray. The single-stranded DNA, which is present on this slide, it has got the property of binding to a complementary DNA. Okay. It can bind. So the DNA, single-stranded DNA in the microarray can bind to its complementary DNA by a process called hybridization. Okay, so this is the property of the single-stranded DNA that is attached to the substrate in a microarray. Okay, so here what we have done is we have taken the cDNA. Okay, so we have taken the cDNA from F1 and F2, and our idea is we want to introduce it into a microarray. Okay, so here what was the process that we did? We used reverse transcriptase to convert messenger RNA to cDNA. And here we have also added, we will also be adding dyes. Okay, so maybe the F1, maybe it is blue, let's say blue dye, and here we have green dye. Okay, so the cDNA is attached to green dye here, and in F2 the blue dye. Okay, then what we do, we mix this. We mix the cDNA from F1 and F2, we mix it, and this mixture we introduce it into the microarray. Okay. So now what will happen is the genes which are complementary to the single-stranded DNA will get attached. Okay, then we will wash it. So the unbound one will be removed. Okay, so whatever is attached, we will find out. Then what happens? The microarray is scanned. So when the microarray is scanned, there are three possibilities. There will be an overlap of blue plus green, that will give yellow. Okay. In some places there will be only blue, in some places there will be only green, and in other places there will be nothing. Okay, so this is the result of a scan. Okay, so the scan shows places where there is an overlap. What does overlap mean? Overlap means F1 and F2 both have genes that hybridize. Okay. Okay, so out of F1 and F2, so out of F1 and F2, who has got the disease? F1 has got cancer. Okay, F1 has got cancer. So here what we are saying, both F1 has got actually cancer. But genes from both F1 and F2 are expressed in an area. So that is not significant in the microarray. Okay. And we said, who has got blue? Blue is of F2. F2 has blue. Okay. F1 is red. All right. So certain areas only the blue is expressed. So if blue is expressed, it means a person with no disease. Okay. That area is the genes which are expressed in a person having no disease. Okay. They express blue. Whereas not red. F1 is green. F1 is green. Okay. So the person with cancer will express green genes, meaning he will have lots of green genes that hybridize in the microarray. Okay, so from that, what information do we get from this analysis? We find genes which are expressed in both healthy and diseased. Okay, these are the yellow portions. Okay, and then there are genes which are expressed only by the healthy. This is the blue. And then there are genes which are expressed only by the diseased. And that is green. Okay. So once we know which genes are expressed by the diseased person, then from the microarray we can locate the position of the gene in the chromosome. Okay, because it is complementary. Okay, so if there is a gene in the microarray, and let's say this gene is from chromosome 1, okay, and the diseased gene that it has hybridized, in this case it is now the green, so this is the one from the diseased person. Okay, it has hybridized to the chromosome gene from chromosome 1. So if it has hybridized, we say that this gene is located at chromosome 1. Okay, so this gene from the diseased person is located at chromosome 1 because it has hybridized to a known location in the microarray. Okay, so initially when we took the messenger RNA from the diseased person, it contains messenger RNA for all the genes that are expressed. But when we did the microarray experiment, we found out that the specific gene in the diseased person is this one, and that we have identified using the microarray. Okay, so now what information have we got? We have got the position of the gene in the chromosome. Now, Position of the gene, how do we know? We also, because there are markers, and that information we get it by analyzing the microarray. Okay, so we have got location. Okay, we can do another thing. We can take the gene from the healthy person and from the diseased person, and we can do sequencing. Okay, so this is one thing that we did. The second thing we can do is sequence the genes, this particular gene. Okay, so the first one, position 1, that gene. How can we sequence it? We can design a primer for that particular gene in chromosome 1. And then we can design this same primer can be used to amplify the gene in chromosome 1 of the normal person. Okay, and the diseased person. So diseased person, we call it the mutant, and normal we call it the wild type. So based on this information, we can sequence, and then we can do blast. We can blast it. What is blast? We are going to overlap these two sequences and see whether there is a change. And if that blast reveals that a particular location there is a change in one nucleotide, okay, then we have identified the mutation. Okay, so we have identified the mutation. Now we have to check whether this mutation results in a phenotype. Okay, so the mutant is a genotype, and whether it causes a disease. Okay, if it causes the disease, we say it is a diseased phenotype. Okay, so we do this also. Okay, so then what do? We look, this is the second thing we can do. Okay, we have already written second. Okay, so the third thing that we can do is we can now go back to this gene. From the database, we can see which protein it affects. Okay. So from the sequence database, we can understand which protein is secreted by this particular gene. And we can find the structure of that protein. Structure of that protein. Okay. So this we can do in the wild type, the normal person. Okay. You can go to the protein, you discover the structure of the protein. And then, based on the structure of that protein, we can locate where the mutation is. Okay. So we can locate the mutation in the structure. Okay. And if that mutation is in the functional site, then it may be causing the disease phenotype. Okay. Suppose that protein is an enzyme, and the diseased person in this particular cancer is overexpressing. Okay, so there can be structural defects or they can be functional defects. There can be overexpression, meaning the person is expressing lots of protein. Okay, so if the person is expressing lots of protein, we know the structure of the protein and we try to find out how we can block the enzyme. Okay, maybe we can develop a small molecule, okay, which can inhibit the enzyme. Okay, and suppose the mutation results in no or zero production of protein, then we can try a gene delivery approach to replace the defective protein, and that would be gene therapy. Okay, so what we are telling is the mutation can result in two things, either overexpression or underexpression. So if it is underexpression, our therapeutic strategy will be to deliver a gene. If it is overexpression, we can develop a small molecule inhibitor. Okay. So all this information we can use to design appropriate therapy. Okay. So that is why we need to discover targets. Okay, so the next question that we are asking ourselves is, okay, what is the importance of knowing the drug target? Okay, we have explained, if we know the target, then we can develop a therapeutic modality. Okay, and that is the importance of knowing about the target in drug discovery. Okay. That is the importance of knowing the drug target. So we can invent interventions. So if there is overexpression, we can invent a small molecule intervention. If it is underexpression, then we can think about a gene therapy, which is aimed at replacing the defective gene. So that is the importance of knowing the drug target. So we have talked about modern approaches. Okay. So there are lots of approaches to discover drug targets. Okay, so one approach usually adopted nowadays, we have tried to explain it here. Okay. So in this discussion, we have tried to answer a few questions. We tried to answer what are receptors. We have defined the receptor. We have asked ourselves the question, why are they important? And we have discussed how are they discovered. With that, let us conclude this discussion.